are starting them with San Diego, CCA San Diego. So everybody please welcome CCA San Diego.
Hello everyone, my name is Olivia Shaw. I'm a senior at Canyon Crest Academy, a public high school in San Diego.
I'm a sophomore at Canyon Crest Academy.
And this year we work to utilize a polysystronic system for enhanced bioremediation.
Okay, so some background. The National Groundwater Foundation states that the hydrologic gradient equals 51% of the drinking water, 64% of irrigation water, and is necessary for many industrial processes in the United States. Therefore, it's very important. But pollution in groundwater contributes to many illnesses throughout the United States because many pollutants can seep into these unconsolidated or unconfined aquifers from landfills, oil spills, runoff in cities, fertilizers, and unkept wells. And this pollution is obvious, very important in the U.S. because it has caused like flood in Michigan, for example. So one solution to this is bioremediation. Many scientists have used this. It uses engineered bacteria to catalyze reactions to break down pollutants within this groundwater system. This can be done in an in situ or ex situ method, depending on the spill and plume. Our focus of our project was to do this in an in situ method, which is on site. So pollutant plumes have a relatively low velocity through porous gradients, which allows bacteria to catalyze reactions in contaminated areas. Therefore, when we place the bacteria in the contaminated area, we are able to catalyze reactions to reduce pollutant effects in that area because the velocity is very slow. So we're able to do this on site. And the way we destroy this bacteria is through another remediation technique called thermal remediation, so it works very well within hydrologic gradients.
Our goal this year was to use systems and enzymes found within the liver to enzymatically catalyze the breakdown of pollutant compounds typically found in the hydrogeological gradient. First, we plan to use a bisystronic plasmid, CYP2E1, and NPR plasmid, for the expression of the cytochrome P450 detoxification protein. The CYP2E1 gene is found within liver cells and breaks down acetone, alcohols, and other biological contaminants. The NPR gene, or NADPH P450 reductase, is necessary for the CYP2E1 expression. We also plan to utilize the ALDV gene already existing within E. coli to enhance the detoxification abilities of our first bisystronic construct. Finally, we plan to purify and isolate the plasmid and implant it into a variety of biological systems.
So for our biobrick application, we verified, amplified, and constructed the bisystronic CYP2E1 NPR plasmid expressing the gene CYP2E1 and the NADPH P450 reductase using ligation and substitution transformation. We documented the application of the CYP2E1 part on the part main page of our wiki as a bronze medal requirement in contradiction to our judging form, and are working to fully sequence this plasmid. The NADPH P450 reductase is essential for the electron transfer in the CYP2E1 expression reaction, so the bisystronic construction allows for a complete expression of the bioremediation CYP2E1 gene. The construct, as shown in the picture, the construct of the CYP2E1 in the NADPH P450 reductase creates this bisystronic plasmid of the CYP2E1 NADPH P450 reductase construct.
We also work towards editing the NADPH P450 reductase gene, typically found in humans, to be RFC compatible and eligible with the biobrick standards. In order to do this, we need to create a bridge that filled the hindrance site of the NADPH P450 reductase to the spell site on the PSB1C3 backbone. We created a bridge with complementary oligonucleotides to create artificial sticky ends with a poly-A tail between the overhangs.
And you can see in this picture how pretty the bridge. So for our results, we will work towards the expression of this bisystronic CYP2E1 NADPH P450 reductase in the E. coli, in the strain DH5 alpha. The This is because this strain of E. coli has an ALDE gene, which will be a co-function of our already phytostonic plasmid. This gene catalyzes a reaction for the metabolism of aldehyde groups created by the catalytic reaction of the CYP2E1 with ethanol groups. So therefore, we constructed a synthetic bacterium for enhancing of the bioremediation processes and functions.
We also work to isolate the NADPH P450 fragment and identify which restriction sites to cut out. As you can see on the picture above, the shorter fragment, or the fragment closest to the bottom on the left too, represents the NADPH P450, and the longer fragment is the rest of our biostronic construct. In the future, we hope to use this to ligate the bridge we created in order to connect the restriction sites from the HIN3 site to the spell site on the backbone. In the future, we want to pursue farther work in validating the functionality of these parts in bioremediation. We plan to experimentally test the effectiveness of these genes in detoxifying ethanol and other organic compounds that are typically found in groundwater. We also want to work with other cytochrome P450 enzymes to create a combinational bioremediation method using bacteria, all expressing different P450 metabolistic enzymes. We will also work with co-expressing the purified NADPH P450 reductase with other P450 enzymes that require this gene for successful expression.
So for our collaboration, we worked with the Nanjing NFLS team from China. And what we did was using chat, we discussed and shared over the biobricks, debugs, constructs, and characterized other parts of our project. So we thank them.
We also wanted to reach out to the community. So we are currently coaching elementary school students in the Write It, Do It Science Olympiad event and we're preparing them for the competition in February. We hope to apply our knowledge of the scientific methods to our project to help teach 4th, 5th, and 6th grade students and encourage them to follow a career path in research and science.
So in the safety of our lab work, we worked in a BSL-1 lab, which is what our school used. We received training from our PI of handling hazardous substances. We reviewed proper safety precautions, how to deal with accidents, and how to properly care for lab materials. The lab required an instructor to be present at all times, and we wore proper attire during our discipline.
We would like to thank Cymer, Canyon Crest Academy Foundation, and New England Biolabs for their sponsorship. We would also like to thank Professor Frederick Lungrich at Vanderbilt University and Professor Chul Ho-Yun at Cheonam University in South Korea for their academic mentorship. We would also like to thank Dr. Ed Gerson and Mr. Ariel Haas and Mrs. Erin Eddingfield for their support throughout the project.
So thank you so much for listening. We hope to see you next year.
All right, thank you. Is this your first time? Is this the team's first time at iGEM?
This is my first time. I think it is.
Yeah, this is our second time.
Second time? Well, welcome back. All right, so we're going to start questions with the judges, and then if time allows, we'll have questions from everybody else.
Thank you for your presentation. It was a very interesting topic. I have a question about the used engineered bacteria you mentioned earlier from the background. You were talking about destroying bacteria. Do you have any?
No, not just destroying groundwater toxins. So, for example, one of the government, what they targeted was they were studying mostly tooling and in the groundwater gradient. So they found a system to basically degrade that in the groundwater system. What we were focusing on was more of acetate groups and ethanol groups.
So after you do that, what is your plan of using those used bacteria?
Yeah.
What's your plan?
We already placed it in the groundwater. So what most scientists do is once you placed in the groundwater, they have this other remediation technique called thermal remediation. And what you do is you destroy the bacteria that you put in the groundwater so that they cannot do any other damage that so it continues. But they're really stationed because when you think of groundwater, you think of a river underground. It's not like that. It's porous medium.